So we are going to talk about photosynthetic organisms, in this case some bacteria. The images are shown here. And typically these photosynthetic microbes are called algae, and we will talk about algae quite a bit today. So here's the topic, biofuels and photosynthesis. First thing I want to do is to alert the two of you and the rest of the folks here about the ICARE's website. That's an important one in my opinion, because that's where you will get a comprehensive view, we hope, of many things that are going on at our own institution, all centered around advanced renewable energy and sustainability. ICARE is an umbrella organization for the entire institution, and under this umbrella, various large and small initiatives that are taking place, large and small just by size, not by importance. And if you go there, you will see that those are listed here, and if you click on those, you can go to the respective sites. The events are shown here. We try to keep it updated as much as possible, and today's highlight, if you go there, you will see that this certificate program is shown. Okay, so my task is to talk about biofuels and photosynthesis. In my mind, those two are synonymous simply because I've been working on photosynthesis all my life. Here is the basic process. Key part is sunlight, light, carbon dioxide, and evolution of oxygen. So this very simple reaction that we are very familiar with, that's at the heart of photosynthesis. And it has some really redeeming features, so I'm going to spend a minute on this. First of all, as is shown here, in the water there are algae, microbes, and on the ground are plants. One of the most amazing things is that they do the same thing as they do. And by that I mean that this process evolved way back billions of years ago on this planet. Abundant source of electrons is this molecule water, a byproduct oxygen, which is good for all of us. But for today's purpose, the two factors, two nutrients that I'm going to talk about are light and CO2. In fact, I will talk very little about light. Those of you who were present at the inaugural lecture by Bob Blankenship last week, he talked a lot about light. I'll touch upon a little bit of CO2. Again, that's a topic that was covered in the last lecture in this series by the chapter Bob. Okay, so this general reaction, it's amazingly green, not because of the presence of chlorophyll, because of what we call green nowadays. It's capture of CO2, production of oxygen, production of good stuff that's good for the rest of the biosphere. All of that is encompassed in this simple set of reactions. Now, over the last few years, maybe the last 5 to 7 years, there has been a resurgence in scope periodically. This is after probably 30 years or so that we now have a resurgence of interest in alternative forms of energy, and that's what we are discussing here. And bioenergy or biofuel became a topic of great interest right around the beginning of this century. And I'll show you two pictures from this popular National Geographic magazine, October 2007. The first one talks about green dreams, and basically here is a warrior, and behind this warrior are very tall plants. Okay? Back in the backdrop are the conventional crops. Imagine that's maize, imagine this is miscanthus, and the whole idea here is that photosynthesis in these plants are doing something great, capturing CO2, capturing light, producing something, and that something in this case is the material that we don't know how to use very efficiently, and that's the lignocellulosic material or cellulosic material. We hear about this quite a bit, a lot of interest, a lot of challenges. We do not know many of the things that need to be done in cracking the code here, namely that material that's called lignocellulose, how to break that open, make the molecules that make lignocellulose, namely the sugar molecules or glucose in this case, those molecules to be accessible for daily use for energy production. That's a big challenge. Lots of activities that's going on. Here at Washington University, ICARES has funded a number of pilot projects that have now, some of them have now evolved into externally funded projects. I'm not going to touch on this today, but this is an important part of the whole equation. This is, again, bioenergy, biofuel, and photosynthesis in the same context. Rather, what I'm going to do is talk about this process, which is use of microbes or algae. And let's start from this very basic picture again. This is, again, the same issue of National Geographic, and it captures the essence of what people are thinking about. First of all, you grow algae, and you grow them in reactors, in this case, reactors that are not very expensive, just plastic bags, and they have to be in Arizona because that's where all the sunlight is, apparently. And behind that is the picture of a power plant. And this is an important concept. The concept is that this is a point source of the food for this algae, which is CO2, and the other food is coming from light. Both are abundant, so one should be able to utilize both of those to grow tons and tons of algae. So written down here is something that probably not everybody can read, so I'll read that for you. Power plant exhaust that could so this algae can soak up carbon dioxide. Algae here are fondly called the green scum, and that's the way at least my grandmother knows algae. So could soak up carbon dioxide while cranking out 5,000 gallons of biodiesel an acre each year. But then it says, at least in theory, in 2007. And in many ways, what I'm going to talk to you about today are the attempts that all of us collectively are making to convert that theory into practice. All right, so again, coming back to this issue of what is biofuel, and a couple of years ago, we had a conference here, and Steve Mayfield from San Diego came here and made this presentation. It's a nice slide directly from his presentation. It says that biofuels are just nature's way of turning sunlight into gasoline. Okay? And one path is this, photosynthesis in crops, and then finally ethanol, or photosynthesis in algae, finally gasoline. Looking at this, let me ask you, especially the students who are taking this as a certificate course, what do you think Steve Mayfield works on? Plants or algae? The picture should give you a sense. Anybody else who doesn't know Steve? Oh, come on, you can make a guess. Algae for sure. And so there have been now these camps, right? One camp that says that lignocellulosic is the way to go. Another camp that says algae is the way to go. Many of us, though, already know that those are not the kind of even discussions that one is worth having, simply because we need all sources of energy, not just biofuel, but also solar, geothermal, things like that we have been discussing. So both paths are going to be useful. All kinds of nice phrases that have cropped up in the last few years. Ethanol is good for drinking, not driving, is one of them. And there are all kinds of things about algae also. I'm not going to talk about those now because I'm going to talk more about algae. So we won't need to be serious about it. Okay. Now, one of the things that algae can do naturally, some strains of algae, that they can make tons and tons of oil, these natural oils, right? And this caught people's attention many years ago. There have been a number of attempts to take that farther, to make lots of oil from these naturally occurring strains. And the idea is that from those natural oils, you can even go up to gasoline. So the concept here then is the algae as the sustainable sources of biofuels and biomaterial, as Rich Axelbaum mentioned last week, that it's sustainable simply because it eats CO2, so it's a carbon negative process. and then you can make biofuels and biomaterial that you can use, so it does not do too much harm to the environment. Now, again, about 2007, there was a white paper that came out from mainly work done by Dave Daggett at Boeing Company, also folks at NASA's a group at Munich, US Air Force. They all got together, and in that case, they were saying, well, you see, you cannot really use any other form of fuel to fly your aircraft. You really need to have something like kerosene. Where can we get this from? And this continues to be a big issue. And it was identified, algae were identified as wonderful sources of that kind of fuel, right? So that's the bottom line. But what they said is that one promising feedstock is algae. And the reason they're saying this is because if you compare it with, let's say, soybean, which grows in the Midwest or all around us in large quantity, algae can produce 150 to 300 times more oil, right? And with the potential for algae providing 10,000 gallons per acre per year, 85 million gallons of biojet could be produced on a land mass equivalent to the size of the University of Maryland, they like Maryland, or not. Moreover, if these biojet fuels are fully compatible with legacy aircraft, then we can supply the present world's fleet with 100% of the fuel needs as well into the future. That's the dream, right? Now, where do these numbers come from? And they come from here. Basically, if you look at the productivity, the oil production capacity of different crop plants, highest here is oil palm, okay? You get certain numbers. And the important thing is this one. If you take all the cropland in the US, what percentage of that needs to be used for all the transportation fuel that is necessary, right? And this is not possible, right? We need another planet or something. We go try to make oil from corn. With soybean, very difficult. We have to invade some other countries to do that. Oil palm, half the land, not so great. Microalgae, numbers are looking better and better. 24, 5, 2.2. That's really great. That's the one that tells you it's Maryland, right? Now, what's hidden in it? What's hidden in this are these numbers that come from very small scale experiments that have been done, where one has extracted or ascertained how much oil is produced by algae, and then you multiply by a factor. And that multiplication, that scaling, I will submit, is the most important issue right now, and many folks are looking into that, including students and faculty from the Energy, Environment, and Chemical Engineering department here. But basically what they're saying is that, okay, now we know that microalgae, 30% oil production is possible, 10 grams per meter square per day of production of biomass is possible, and in that case, we'll have to use 24% of all the land in the US, okay? And then in a different color I show, if you can increase this 10 to 49 grams per meter square per day, then that number goes down to 5. And finally, if the 30% oil goes up to 70% oil, and then you get 2.2, okay? So in the different colors of things that have not been achieved yet, need to be achieved to fulfill this dream. And today, we will discuss how that can be done. Because we talk about this all the time, and one of the first things that happens is that somebody talks about this, and it gets on the newspaper, especially in St. Louis. I've seen many times these assessments that come out in the local newspaper that says, we are going to solve the problem. And if we cannot solve that problem in a short period of time, then along with that comes some form of despair. Perhaps this is not going to work. And our job as scientists and technologists who are involved in this kind of work is to keep everybody, aware of, make them aware of what can be done today, what are the potentials. Don't try to oversell. And so let's look at all of those things today to see where we can take this to. All right, so again, advantages of algae. Non-arable land like deserts, algae grow everywhere. Yields up to 10 times those for land plants, again, the same kind of thing. This is an important one. You can use wastewater to grow algae. It can grow in the kind of water that cannot be used by crop plants easily. Non-fuel fraction is high in protein and edible. Those are the co-products that can be used. Capture CO2 at point source, I've talked about this, and can produce high use of energy-dense molecules. Those are the fuels. Okay. So here at Washington University, we have at various places collection of algal strains, and one such collection at iCares has, at that time it was 28, now we have many more algal strains of different kinds that are present, and this is kind of a resource. People who want to use this can use these strains. Let's talk about now the concept of algae as factories. Okay? So far what we have talked about is algae as the source of biomass. A lot of algae that you need to produce. But another way to look at it is kind of like, if you're a connoisseur of wine or beer, you know that you use microbes, not to make a lot of microbes, but to make all of the value-added products like beer. So in that case, you are using those microbes as factories. And that's the same concept here. Can we use algae as factories? And I think there are experiments that are going on in different labs, and some that has been scaled up says, yes, you can. So here is a diagram that says, here are the energy-rich compounds from microalgae, and photosynthesis is at the heart of it. I'm not going to go into the detail of any of this. And photosynthesis leads to fixation of carbon, CO2, comes in and gets fixed. And from that, all the magic that happens inside this algae, one can produce different kinds of compounds inside the cells. And those compounds can then be converted, because we are using these algal cells as factories of all this good stuff. So the inputs are simple things like water and CO2, some nutrients. Those are important. You can also supplement the CO2 and light with other carbon sources that can enhance the productivity of algae. And this is an approach that is being used by companies such as Solazyme now, where they are saying, here is algae that can make something, and we have a very low-cost source of carbon, namely sugar. If we add the sugar to this algae, what can it make? And they can make a lot of oil in using that kind of an approach. So these are the inputs. And the outputs are all of these. All of these are possible, and most of this has been shown that can be produced by algae. Okay? So algae as factory, here are the energy-rich compounds that can be made by algae. I'm going to switch over and talk about some of the work that's going on in my own lab. And here is probably a little bit of data, just to show that the potentials are enormous. Well, the first thing is that algae can grow just about everywhere on this planet. And as a result of that, they have tremendous diversity in their metabolism, because different kinds of algae, they have learned how to cope with different types of environmental conditions. And as a result of that, they are very rich in their metabolic repertoire, as it is called, or the ability to make different kinds of compounds. So here I'm showing you cyanobacteria, which used to be called blue-green algae. Those are the ones that we study. By all means, they are algae, but we do not call them algae nowadays. They are cyanobacteria, and they produce oxygen and they use photosynthesis. They come from a wide variety of habitats. Morphologically, they're diverse, metabolically complex. This is what I just mentioned. And here are some pictures. What is shown here are these cells that are 2 micrometer diameter. That means they are small. This one is 4 micrometer. This one is in a filament, anabina. Each cell in a filament is about 2 micron. Whereas look at this one, it's 10 micrometer. That's big, right? So they come in all kinds of shapes. And we have been studying these organisms in great detail. Notable features of cyanobacterial metabolism, I've already talked about photosynthesis, CO2 fixation. Cyanobacteria are also endowed with, some cyanobacteria are endowed with the ability to fix nitrogen. And that's a good thing, because we really need to replace the fertilizers that are chemical in nature in the near future. And so the idea also is that if you can use this kind of cyanobacteria, they can not only act as a resource for energy-rich compounds, but they can also provide the nitrogen sources that are necessary to ultimately grow plants and other things. Okay, so nitrogen fixation is an important one. And then for biologists here, and a very important characteristic of cyanobacteria, actually the only known bacteria that have circadian rhythm just like you and me. So they experience jet lags, and I will show you some data that tells you how they can utilize this kind of a rhythm to do all kinds of interesting metabolism, to make all kinds of interesting compounds. In the era of genomics, which is what we are in, that means you take an organism and determine the sequence of its genome. It used to be very expensive. It used to take a long time. And somebody like Gautam Danta sitting here can probably do the bacterial genome sequence in an evening over a glass of beer. That's the situation right now, right? So that means this is very easy to do, and you see that because nearly 100 cyanobacterial strains have been sequenced. And the sequence tells us, again, the metabolic capacity of each one of these organisms, of what are all the things they can possibly make. It also tells us what are all the things they can possibly not make, but if we can engineer them just a little bit, then they can make those things, those desired products. So genome provides us with the map that we need to understand the organism in a deep manner, and also allows us to then engineer it further so that we can make the desired products. That's why I'm bringing in the whole concept of the genome sequence. So this genome sequences then provide the blueprints for the diverse metabolic potentials of these oxygenic bacteria. Here is what the organism looks like under an electron microscope. They look like this. In the lab, when they're growing, they look like this in bottles. These are small-scale experiments in our labs. In this organism, there are some notable features, and let me point those out to you. Inside the cell, this is a bacterium. Inside the cell, there are these special membranes. Those are called thylakoid membranes. That's where photosynthetic electron transport reactions occur. And then there are inclusion bodies inside the cells that are very important. And the one that I want to point out to you is glycogen granules. These are glycogens just like in you and me. Basically, glucose molecules are stored in the form of glycogen. And what we will see in a minute is that these organisms know how to use photosynthesis in the daytime to make lots and lots of glycogen to store away the energy. And then at nighttime, when photosynthesis is not occurring, utilize that glycogen to derive the energy and create special conditions under which all the reactions that require the absence of oxygen can take place. What I mean by that will become clear as I go on. Okay, so glycogen granules are important. Cyanotherapy can store and later mobilize the fixed carbon. And here is the cartoon diagram that says, okay, you do photosynthesis, make glycogen, and at the night time, this is in the daytime, and at night time, you can just utilize this glycogen, and then from the energy that's derived from that, you can do lots of cellular metabolism. One of them is fixation of nitrogen. This reaction that's mediated by this enzyme called nitrogenase, and we'll talk about that in a little bit more. This enzyme is unique in the biosphere in its ability to fix nitrogen, and it's one of the two enzymes known that can also help in production of hydrogen, right? And hydrogen is a good energy carrier. Biological production of hydrogen has been looked into in great detail in the past, and we have spent some time in examining the potential of these organisms to make hydrogen. So hence I want to point that out. Okay, so how does that happen? It happens concomitantly with fixation of nitrogen. This is again very simple basic reaction that we have known for a long time. And basically nitrogen is fixed in the form of ammonia, and hydrogen is produced. So in this case, in this reaction, as you see, for every molecule of nitrogen that's fixed, you produce a molecule of hydrogen. Okay? You don't have to do anything special. It just comes out automatically. Okay, here's a statement that I made, and I want to dwell on this a little bit more. Temporal separation of nitrogen fixation from photosynthesis allows nitrogens to function under suboxyl intracellular conditions created during the dark period. What does that very long sentence mean? One of the problems that one faces in using algae is that algae makes oxygen, right? Why is that a problem? Because most of the processes that we usually deal with, the most important one is making beer, of course, is fermentation. And that's a process that requires absence of oxygen. Fermentative processes are the ones that do not like to have oxygen around. So they take place under anoxic condition or low oxygen tension condition. Similarly, the nitrogenous reaction also requires low oxygen tension. If there is any oxygen present, not only that the reaction will stop, the entire apparatus, the enzyme that fixes nitrogen and makes hydrogen, falls apart. This is the conundrum then. How can you fix CO2, derive energy from light, evolve oxygen, and in the same organism, in the same cell, also produce hydrogen? And there are lots of different ways that people have been studying this, lots of different approaches, but all centers around how do you take care of the oxygen that's being produced. Various organisms use different strategies. This one is very clever. What it does is every day it fixes CO2, evolves oxygen. Every night it has a wonderful machinery, mainly based on respiration, that takes care of all the oxygen. It just scrubbed off. And at night time, then you fix nitrogen. Excuse me. Now, the other side of the coin is nitrogen fixation is extremely energy dependent. And that's shown by the 16 molecules of ATP. Okay? It's extremely highly energy dependent. So where do you get the energy from? The energy comes from this. So now you can see the pattern of what's happening. This bug, the same cell, in the morning, it fixes CO2, evolves oxygen, and stores away that CO2 in the form of glycogen. At night time, it has high rate of respiration, takes care of all the oxygen. And then the glycogen is degraded, and the energy derived from that is utilized, in this case, for nitrogenous function. A beautiful system that goes back and forth, day and night, doing this thing periodic way. And the circadian rhythm that I talked about, the day and night cycle, helps these organisms to do that. Okay? So the bottom line then is, here is, from this vast range of different kind of algae, we decided to study one in which every cell has this ability. This is a very unusual thing. Not all algae can do it. But by taking advantage of this, we figured out the true potential of this kind of algal strains in the production of hydrogen. And let me tell you what that is. That's not good. All right, we need that. All right, that's good. So here are some standard algal strains that have been used in the past. And I'm not going to go into any details other than just showing you these numbers. Okay? So in some units, doesn't matter what that unit is, here are the numbers: 6.6, 1.2, 45.2. Here is another number, 0.92, almost 1. Okay? Here are the different bugs. Some of them are oxygenic, most of them are algae. Here is one that's an anoxygenic bacteria. Right. The other thing to show is this business of aerobic incubation and anaerobic incubation, right? So the question is, can you make hydrogen when you are taking cells or algal cells under normal conditions, where you do not have to create special anaerobic conditions? And what we found out is that Cyanobacterial, this special alga that I just talked about, this is what it does. Here are the numbers. And without going into any more detail, you can see that the numbers are much higher than what has been looked at over the last maybe 20 years or so. And why is that so? We have all kinds of reasons why, that we can provide. But the point is that among all of these algae, here's one that does this special thing, and it can produce hydrogen at rates that are much, much higher than what has been determined in the past. That's the bottom line here. So again, the whole business of the metabolic potential of these algae. We went into this in much more detail, and maybe I will not go into all of those. I've already mentioned these are the enzymes in biological nitrogen fixation that are important. And we determined that in cyanothesis, the nitrogenase that's doing that, not hydrogenase. And nitrogenase is doing it the way that I have already mentioned to you. I'll not go into the detail, but this one is important. And this one is important because of this business of CO2. When the cells are grown, these algal cells, under aerobic condition, they produce this much hydrogen. When you add a lot of CO2, and in this case, lot was 8%, which is half the amount that you usually find in flue gas coming out of a coal-fired power plant, under those conditions, see, this number jumps. And that number is very similar to when the cells have added alternate carbon source, which in this case is glycerol. So that's the point that I want to make, that the organism itself is already tuned to what we really desire from them, which is you add more CO2 and you get more product. And that's what's happening. And you don't really have to have glycerol. You can just have CO2 coming from a point source of CO2 production. And this side actually says that basically what's happening in these two cases is the amount of glycogen that's stored that goes down, and as a result of that, you get hydrogen production. Okay, and again, this one shows again that we tried it various ways, and without making mutations and things like that from this alga, we can make tons and tons of hydrogen. And I debated whether I should show you, when we published this last year, and this work was supported by Department of Energy, as well as Consortium for Clean Coal Utilization here at WashU, which is under the umbrella of iCare. This work was then immediately caught people's attention, because it said that you really do not even need to do genetic engineering. The organism that you can find out in the nature already has the capacity to do this. Okay, let me now move on to the next part of the story, which is this whole concept of using algae to sequester CO2. And I want to discuss this because here again, there are all kinds of stories that are going around. Some people think that it's impossible to use algae to sequester all the CO2, me being one of them. And there are people who are really looking forward. They're saying, why so? You know, algae are the one who, for the longest time, have sequestered lots of CO2 and do photosynthesis. And it's possible to do that if we take an approach where we go to really large scale, not really going to, 10,000 liters or so, but the entire ocean or all the oceans. What happens then? And again, somebody named Yusuf Chisti from New Zealand came to visit with us during that meeting that took place 2 years ago, and he made that calculation. He's a chemical engineer by training, and he made some calculation. I want to share that with you. And this is again so that if you, the students here, if you are faced with this challenge, saying, can algae really sequester all the CO2? You know how to approach that. So let me share that with you. Okay, so the CO2 capture by nutrient fertilization of oceans. The concept is that nutrient poor oceans are nutrient poor. And if you then add specific nutrients, and without going into the details of those, there have been many studies that have been looked at that, then you can get what is typically called an algal bloom. Now, bloom is something that people associate with algae all the time, okay? Again, if my grandmother were here, she will know algal pond scum, and she will know algal bloom. This is what everybody knows about algae. Now, hidden in that is the fact that really under certain conditions, algae can grow at a very high rate and over a large body of water, right? So CO2 in atmosphere then gets converted to carbon and biomass. And in that case, you don't want to utilize that carbon. You just want to sequester it. That's the concept. So what does it mean to do that? And in the open, man-made algal production system, raceway ponds, the typical biomass productivity, here are the numbers, okay? 25 grams per meter square per day. And let's walk through these calculations so that we understand what it will take to sequester all the CO2 that is man-made. And I understand that there are still debates that, that man-made CO2 may or may not making changes in climate. There's a division in opinion. I belong to the group that says yes, it is. And so if we have a way of sequestering their carbon, what would it take? How much algae do we need to grow over what percentage of this planet? So culture conditions, important. Carbon dioxide supplemented culture from point sources. Equatorial location, clear skies, lots of sunlight. Nutrient sufficiency, you have to have these things. And the studies that are being made in this Department of Energy and Environmental Chemical Engineering, it has been found that some of these elements are extremely important. They are expensive and also important for the growth of algae. And then this is another business, 0.25 meter deep pond. You cannot go too deep, and this is something that Bob Blankenship touched on last week when he talked, because you don't want light limitation. And I'm not going to talk about light, but these are the culture conditions. Okay? So under this case, CO2 sequestration in the ocean, and let's look at the numbers. The best case scenario, right? You have everything. Then you will have 25 grams per meter square per day, which translates into 91 tons per hectare per year, which translates into 171.4 tons of CO2 per hectare per year. That's the number, okay? Worst case scenario, where the ocean biomass productivity is 5% of race waste. And these are real numbers. There is some relevance. I mean, this 5% number is not, pulling out of a hat. And that one is 8.7 tons. So that's the difference, 171.4 and 8.7 tons. And in this case, here is the stoichiometry. 1 ton of dry algal biomass is made from 1.883 tons of CO2 capture. Okay. All right, so here's what happens. For complete absorption of global CO2 emissions, it's man-made CO2 emission. Emissions from fossil fuels is that many tons. The ocean surface area that's needed for best case scenario is this. That's a very small number. Worst case scenario, which is probably what we should be looking at, is 0.009% of total ocean surface. And since Chisti was originally trained in the UK, he takes the UK, and that's good. We can look at that and we can get some idea. 10% of the British Isles, UK plus Ireland. But we are talking about the entire planet, all of the mapping. So that's not a big number. Now, is that possible? Does that happen? And these are the two nice pictures that he showed that were quite interesting. We had to write down, maybe we can see this, but you can get a sense. What you should be looking at are, it's impossible to see this with these lights on, but I don't know how to turn the lights down. But basically what it is a bloom all around the British Isles, right? And this is captured from satellite image. This is possible. In our country, you may want to think about the dead region right south of New Orleans, where all of the good stuff flowing through the Mississippi River ends up being at, and tons of algae grow, and algae grow to such a high density that it actually, because of the respiratory activity, it takes away all the oxygen that's present and nothing else can grow there. That's why that's a dead zone. Okay? So lots of algae can grow over a large area. And here's another picture. This is the area of the Black Sea. And the picture shows that the entire thing is full with algae, algal bloom. Okay? So area of Black Sea is 138% of British Isles. So now, if we go back to these numbers, and this is the bottom line, 10% of British Isles, all covered with algae, should be able to take care of all the CO2. So if we knew, and that's the challenge, if we knew how to do this in a controlled manner, then we'll solve the problem for the planet of sequestering all the CO2. This is still far away, yes?
Wouldn't we have to do that every year then?
That's a good question. Do we have to do it every year? Does anybody else have an idea? Every year to do what?
To sequester all the CO2.
Right.
And if we're making fuel with
right.
that algae, then we're going to put it into the atmosphere anyway.
Good. So, in your opinion, then if you do it every year, yes, the answer is yes. What needs to happen to all of those algae?
Needs to.
Every year you have so much algae.
Then you need to permanently sequester that algae.
That's right. Absolutely. And so, basically, then you have to have algae that are heavy. They will sequester all the CO2 at the top level and sink and be stored away for a very long period of time. It's coming very close to the production of petroleum and coal, you know. Millions of years later, that probably will give rise to all those fossil fuels that we always talk about. That's how they originated. But that's the goal. You cannot have both things going simultaneously. You cannot have these algal factories which are sequestering CO2 and then doing fermentation and getting rid of all the CO2 out or burning it in the automobile or a power plant. You really need to figure out a way of this algae then sequestering all of this CO2 using photosynthesis sunlight and then let them sink, be stored away, and there will be a fresh crop of algae the next year with a bloom and sequester all the CO2. So there is a way. It's still far away because we do not know how to do it in a controlled manner. But if anybody asks you the question, I think, and we have looked at these numbers very carefully. They are reliable, and which is where the value is. You can remember just this. This much area is enough to take care of global CO2 emissions. Okay?
But it has to be a year-long.
It has to be a year-long, and there has to be productivity. But it's taken care of in this worst-case scenario. Okay? It's not so esoteric in that sense. But the major problem is we don't know how to do these things at this large scale. You know, that's the challenge. So needs and recommendations in this area that came from actually a report that came out a couple of years ago. First of all, low-cost technology to produce microalgae. This is very, very important. Stable cultivation at high productivity, something that we just talked about. Co-products are very important. Biofuels ain't going to do it. Biofuels needs to be produced in high volume, and inherently they are of low cost per unit. So in order to match that challenge, there needs to be something else that's of some more value, and that's where the co-product concept comes in, high value chemicals in this case. Co-processes, such as flue gas capture, wastewater treatment, all of these things will capture the value that's needed to make this thing worthwhile for businesses who will take this challenge. And then you have to have these collaborations between utilities and algae biofuel producers. This is not going to happen by just one group working on its own. And then finally, I guess this is where we come in, right? And you know, all of these things that we are doing here at PARC, at ICARES, here at the Brauer Hall, this collaboration between biologists, engineers, and utilities. They all have to work together to improve the reactor designs for newly engineered organisms and to develop novel uses of algae for energy production. So the story is kind of mixed. That's what I wanted to present to you, is that there are tremendous potentials, we know that, but the tools and the knowledge base are still far away from what will allow us to do all of these things in an effective manner. And that's probably what the education and training and the research in the coming years is going to give us. Okay. That's it. Questions, comments, discussions?
I had two questions. So one was, in the first part of your talk, the Cyanophaceae data. Given the diversity in the algae, the cyano, do you think that that's, you know, you presented that as this isolated example. Is that what you think is going to be? You know, that there are these rare examples of these sort of interesting mixes of metabolic pathways sort of being dealt with, or as people explore perhaps other genomes and sort of these kinds of prospecting of microbes that we might stumble upon?
I don't think that there will be as rare as they seem to be right now. Because over the last 50 years, remember, biology has always gone to a very few but well-studied microorganisms or organisms that we study in great detail. But now, with this explosion in genomics, we go after organisms that we have never been able to even grow in our own labs, right? So I don't think that it's that unusual. And in fact, when we started our study with the first one, we determined that there are, half a dozen that are in different culture collections, and we are continuing to study those also. And they all have the same kind of properties, so I don't think it's going to be rare.
Second question related to the second part, because you said that you yourself don't really believe in this algal solution to sequestering CO2. Is that because solely of the problem that you mentioned, or are there other reasons that you didn't discuss?
No, I don't believe in that for the short term. In the long term, yes, this is possible. But when I say I don't believe in that, that's in keeping in mind that some people say, oh, we can do it today, right? And if you think about it, the whole concept of a bloom, which is something that very few people really understand, and I would submit that very few people know the rules of the game. What do you need to do to promote the growth of the bloom, and how do you control it? Because the worst possible thing will be that you know how to trigger it on, but you don't know how to turn it off, right? You don't want this entire planet to be a monoculture of one kind of algae. That's scary, right? So you don't want to go there. So you have to have the on-off switches understood really well, and then figure out what are all the resources that are necessary to grow it in large bodies of water or wherever. So that's why I'm saying it's going to take time. That's all I meant. Yeah.
How consistent and how large is the bloom you described at the mouth of the Mississippi River, and for how long a duration will it last?
Well, blooms come in different forms and shapes, right?
Right.
Some that are there forever.
Yes.
And the mouth of the Mississippi is one of those. It's just very large. We should actually go into there are beautiful pictures and that take care of this. It's a very large body of water.
I mean, what I'm getting at is, like, if we just increase the amount of agricultural runoff into every tributary in across the nation, and then basically feed the mouth of every major river, we can create these sustained blooms just simply by poor farming practices. And obviously,
does anybody else have any comment on that?
I mean,
this is great.
I know really very little about
right. I mean, what will happen is that our daily intake of seafood will go down dramatically because there will be nothing else growing there, right? And then the other problem with that kind of a bloom is the fact that you can see them for year after year. They're just floating on the top. That's not going to solve the problem, because ultimately these blooms, the biomass gets destroyed in a way because there's a high amount of respiration that starts taking place. You don't want to go that way. The best scenario will be you sequester the CO2 and you get heavier and then you sink. And so I think that, yeah, what you mentioned, that will be one way, but maybe that's not the way that we want to go. Yeah.
So it's related to that heavier idea. I mean, has that been achieved in the lab? I mean, is there enough whatever metabolite accumulation that actually density appreciably changes?
Well, there are all kinds of ways. These algae actually do things. I mean, there are some algae that have inside them special gas vesicles. This is over a very short depth, not huge depths that we are talking about. And it controls the production of those gas vesicles. When you do not have a lot of light, then they make these gas vesicles float up. And when you have a lot of light, this is a very simplistic way of looking at it. Imagine diatoms, imagine other algae that have, that are denser because of the way you are storing it with their CO2. Maybe ones that will take directly CO2 and put it in the form of carbonate of some kind. All of those we know exist in the nature. Maybe some other people in this room have more ideas about this than I do. Any other question? I've been told that there are biofuels, but they are of poorer quality than after Bob's talk. Namely, pizza is not going to be here today, but there is going to be cookie and also drinks. So let's conclude here and go enjoy the biofuels.